Hello everyone, my name is Lena Michailidou and I'm a doctoral researcher at the University of Crete. My doctoral project focuses on how disrupting host factors can suppress viroid infectivity. The work I'm presenting here is just one part of this broader PhD project, focusing specifically on the identification of novel host proteins that interact with viroid RNA using orthogonal organic phase separation. Viroids are small but commercially important plant pathogens. Unlike viruses, their genome is a small circular RNA that does not encode proteins. A few host proteins are known to play a role in their infection cycle, but after more than 50 years of research, we still know surprisingly little about how viroids interact with their hosts. Because of this knowledge gap, our goal is to systematically identify new host factors that interact with viroid RNA and that may be critical for infection. To achieve this, we use a method called orthogonal organic phase separation, or OOPS. OOPS allow us to capture RNA binding proteins directly from plant tissue. The process begins with UV cross-linking, which stabilizes RNA-protein interactions in vivo. The resulting complexes are then purified, separated from other cellular components, and finally released from the RNA for proteomic analysis. Using OOPS, we compare RNA-binding proteins in benthamiana plants infected with potato spindle tuber viroid, PSTVd, to those in healthy plants. Through this approach, we identified 125 RNA-binding proteins that were differentially regulated between healthy and infected plants. We expect that these proteins could bind either to the host RNAs whose level changed during infection or directly to the viroid itself. This provides a much broader picture of the host proteins that potentially involved in the viroid life cycle. From this, we have selected the first 10 proteins for initial study. To test whether these proteins are truly needed for infection, we are now using virus-induced gene silencing, or VIGS. We could say that VIGS is a technique that temporarily switches off plant genes. The method uses a modified virus carrying a small piece of the target gene. When the plant is infected with this virus, its own silencing machinery degrades both the viral RNA and the corresponding host transcript. In this way, the target gene is silenced. By silencing each candidate gene and then challenging the plants with PSTVd, we can test whether its absence affects infectivity. If we find interesting proteins, the next step is to know whether they act directly by binding the viroid, and for this we plan to use RNA or protein immunoprecipitation. Our goal is to identify novel host proteins that interact with PSTVd and to understand how these interactions drive effectivity. By identifying the factors that viroid rely on, we can test how disrupting them suppresses viroid effectivity. This not only deepens knowledge of PSTVd biology, but also highlights potential strategies for developing resistant crops. Thank you very much for your attention, and I will be happy to discuss our findings further during the poster session.